Hello again. Welcome everyone to MACSMIN 2024. Now, Professor Simon Billinge from Columbia University will talk about materials genomics, materials heredity, and the structure definition problem. Over to you, Simon, please.
All right, so thanks very much, Vitaly. It's really a pleasure to be here. Sad that I can't be there in person. I just came back from Europe and I was hoping I could squeeze it in, but our semester has already started and I would miss too much teaching. But anyway, I'm happy to present remotely. Thanks, Vitaly, for having that option. Okay, so some of you were at the MACSMIN last year, and I gave this presentation. I've also given a similar presentation at various places at the SIAM and at various chemistry meetings, so some of it will be familiar to you, but I wanted to actually develop some of the ideas a little bit further and then raise these two questions about materials heredity and the structure definition problem. So it was interesting that we had a lot of discussion today about this topic, the last one, the structure definition problem. So we'll get to that at the end. I think it's actually a pressing problem. So how do I advance my slides? Let's see. Okay, got it. So I'm at Columbia University, and it's in the New York City. This is what it looks like looking south down Manhattan. And I always like to say that there's this big green area here, it's Central Park, and another green area here, this is Riverside Park. And there's a third green area, which are these lawns here. This is actually Columbia University, and I draw it like this. And for those of you who don't know me, I'm a structural scientist, but I'm coming from the experimental end of things. I'm not by training an applied mathematician. And my interest in mathematics has grown over time, and I'm gradually moving in that direction. It's a pleasure that you guys allow me to hang around with you, basically. So I do experiments, and I use high energy and highly powerful sources of synchrotron and X-rays and neutrons. And when I do that, I go to these other labs. So this is actually a synchrotron X-ray source at Brookhaven National Lab, which is 50 miles down the road from here. And this is an accelerator-based neutron source at Oak Ridge National Lab. So maybe it's fun for the mathematical audience to see how experimental scientists do their science. So we take beams of electrons and we put them we make them go in a circle at relativistic speed. So they're going at 99.999% the speed of light. And they radiate X-rays in very intense pencil-shaped beams, and we fire these at our materials. So as well as fun with mathematics, we have fun with electrons. So that's a little bit about my background. And in my research group, we spent essentially my whole career looking at the structure of materials, but not the average structure. So not going beyond the crystal structure, and looking for things like defects and broken symmetries. So that's really the guiding thing in my work. So we study the structure of real materials in action. So we put batteries in beams and we look at how the atoms move around in batteries. So from our point of view, actually the crystal structure is just the starting point. Materials that have the crystal structure in their ground state, they're very boring. They're not doing anything. And when you put them into a battery, you want them to do something. You want lithium to go in and out, and you want these things to happen. So we're actually interested in going beyond the crystal structure, but it turns out that you have to actually have a good definition of the crystal structure to go beyond it. So I also spend part of my time thinking about that. And so we apply our methods to look at materials for sustainable energy, health, and so on. And then recently, as I'll mention in my talk, we've been looking at machine learning and AI applied to this problem as well. So the topic of this talk is really on the materials genomics end. And I'll mention, I think some of you may know this, but it's a very European audience, and this is not so widely known in Europe, but there's this concept of the Materials Genome Initiative. It's a US federal initiative, and I'll mention it in a second. But it's based on the idea of having a genome for materials. So in order to have a genome for materials, we need to understand what a genome is in principle. And the Materials Genome Project basically grew out of the Human Genome Project. So this was a project in the 1990s. It was a US federal project. It involved National Institute of Health and Department of Energy. And they wanted to basically sequence the human genome, and they wanted to make it a kind of a moonshot project that everyone would work on, and we'd solve the human genome inside a decade, something like this. And they succeeded. They threw money at it, and lots of really great science came out. And it was a huge success, basically. Now we've sequenced the human genome, and we can do it really rapidly, and it's turning into therapies for health effects and things like this. So that was the Human Genome Project in the 1990s. So what is a gene? So a gene can be thought of in various different ways. One way of thinking about it is actually as a molecule, a DNA molecule, double-stranded DNA, where you have these base pairs holding together these two ribbons of amino acids, not amino acids, of phosphate groups. And this is what carries the code for life, basically, and it lives inside every cell in your body. But really, another way to think about what the gene is the code itself. So inside that DNA molecule, you have these different chemical species that form into pairs, and then these pairs can be given a letter. And then you just go along this long chain here, going from one point to the next. And each one of these corresponds to a letter. Every three of these corresponds to a codon. And then every codon is then sequenced, and each codon codes for an amino acid. So each triplet of three letters is coding for a particular amino acid, and then you're going to make this long molecule, which is a polymer of amino acids. And then that thing is going to become a protein, and then that protein is going to fold up into a three-dimensional structure. And then the proteins are what give us all of our functionality as organisms. So a third way of thinking about what a gene is, it's any one-dimensional sequence of symbols that codes for a 3D uniquely folded protein structure. So this is my own definition. I just kind of made it up, but it's one way to think about it. So that's what a gene is. So given the success of the Human Genome Project, some very clever material scientists somehow persuaded the US government in 2011 to have a Materials Genome Initiative. So again, it's a multi-agency thing. It involves Department of Energy, National Science Foundation. It doesn't involve NIH this case, but it NIST, other US federal agencies. And they say they got President Obama involved at the time, and they said, okay, if we're going to have another moonshot thing, which is like the Human Genome Project. So the only problem with this, of course, is that you can't sequence the materials genome because materials don't have a gene. So what they really meant by this Materials Genome Project was this is from their original documentation, that they wanted to use genomic methods, including at the time kind of data mining and a continuous integration of theory, synthesis, and characterization to accelerate the discovery of new materials and also the deployment of new materials in devices. In particular, they wanted to cut down the cost by a factor of 2 and the time by a factor of 2 that it takes to bring materials from inception to being actually in production. So that was the idea. Here are all the agencies involved down here. So it's basically a big government initiative. And like I said, the problem is there's no materials genome, but the basic idea is we'll use genomics methods. This was what's going to happen. This axis is time, and this is number of new materials to market. And using the conventional approaches, the number of new materials to market is rising very, very slowly as a function of time. But with our materials genome, it's going to go up like a rocket. It's going to be fantastic. We're going to solve all of our problems in health, sustainable energy. Fantastic. That was 2011. So did it work? Well, the answer is no. Basically, it didn't work. 10 years later, 2019, here's an article that was put together to say how wonderful this had been and how fantastic it was and how successful it was. And then you go and you look at these examples, and there are all these tiny edge cases that have had no real impact whatsoever. They haven't discovered new drugs, they haven't discovered new batteries, nothing. But at the same time, you know, there were things that you could point to. There's the Materials Project, for example, which is a DOE-funded activity at Berkeley Lab. And it has hundreds of thousands of users, and they have these large databases of material data that has been produced by theoretically, basically using density functional theory. And it has 19,000 citations. It has 200,000 registered users. I mean, this is demonstrably a success, no question. So depending how you define success, you know, it's been okay, it's been good. And to be fair, it was a little bit ahead of its time. So 2011, nobody was applying AI machine learning methods. So what they had in mind for genomics was basic good old data mining, things like this. But now we have AI and machine learning. These approaches, these kind of genomic approaches, they're definitely going to accelerate. So this is the basic idea. So 2011 was when it was initiated. 2021, 10 years later, they've renewed it. So the MGI has been officially renewed for another 10 years. They have a new strategic plan and they have a new logo. And so fantastic, we're still in business. We can do this for 10 more years. So given that I have this definition of a gene, which is a 1D code that codes for a 3D structure, I started to just kind of slightly whimsically start thinking about, well, there's nothing like DNA that codes for that can be called a materials genome or a materials gene, but could we come up with a gene for materials? Like, in other words, can we think of a 1D sequence of symbols that codes for a 3D uniquely specified material structure? And if we can, then that can be our materials gene, and either that's a useful idea or it's not a useful, but let's explore it. You know, we're among mathematicians. We're allowed to explore things that are interesting, regardless if they're useful or not. Okay. Well, let's spend a moment thinking about what the current ways we have of coding structure. And it's completely based on crystallography. Not completely, but it's really crystallography centric. So basically, we want to take a crystallographic viewpoint where we define a crystal structure. This 99% of structure stuff is basically done this way. And we know how it works. We have crystal systems, we have space groups, and then that we have on the code side and on the real space side, we have a unit cell, and then we have some kind of mapping of structures into the unit cell, and we apply the symmetry conditions to generate the structure. So it's a good thing. It's a symmetry forward. You start with symmetry, and you define crystal systems, and then Bravais lattices, and then you define space groups, and then you define a unit cell, and then you define an asymmetric unit, which is a structural motif, and then you wrap it all backwards. You then apply the space group symmetries to the asymmetric unit to generate the structure, and so on. So it's a good coding. I say that it's a symmetry forward. We think that symmetry is really the most important aspect of the structure. And so what are some of the strengths? Well, it's been around for 100 years and it's influenced many, many thousands of people. Why is it so successful? And it's one of these reasons is that it's symmetry forward. And if you know the symmetries, even if you don't know the structure, you know something about materials properties. if your material is centrosymmetric, you can't have certain properties like piezoelectricity and so on and so forth. So there are some material properties, and let's face it, as chemists and material scientists, we don't care about the structure, we care about the properties, and the structure is just a way of getting to the properties. So knowing something about the symmetry tells you something about the properties, but it honestly doesn't tell you much. Like if you know the symmetry, it doesn't tell you if that's a metal or an insulator. But there are some properties that depend on symmetry. Another strength of this coding is that incredibly complicated structures with 10 to the 23 atoms in can be reduced to a really tiny set of parameters, like 10 or 15 parameters. The lattice parameters of the unit cell, the symmetry operations, the positions of the atoms in the unit cell. Incredibly efficient coding. Another strength of the crystallographic coding is it's extremely well aligned to the way that structures are solved. And these two went kind of hand in hand in a way. When the X-ray crystallography was developed, space groups and crystallographic representations were it was a co-design. They grew up together. And basically, when you do an X-ray diffraction measurement, you get Bragg peaks, and the positions of the Bragg peaks tell you about the unit cell. The systematic absences of the Bragg peaks tell you about the space group. And so there's a very close mapping between the way that these structures are measured and then this coding. Another strength and one reason why it's been around. And I think everyone on the call will agree that it's allowed us to become very mathematically rigorous. And in fact, space groups and matrix representation theory is behind a lot of quantum mechanics. And I think, I'm not 100% sure about the history of this, but I think that, again, it kind of grew out of this crystallographic development. So really powerful concepts and very powerful mathematics came from this. And then finally, another reason why it continues to be around and it won't never go away, at least for the foreseeable future, is that the whole generations of crystallographers and chemists have basically been taught this way. And so there's a huge inertia. If we want to change how we do it, we have to overcome this incredible inertia. So these are strengths of the crystallographic coding. What are some of the weaknesses of the crystallographic coding? And this is stuff that everyone on this call, I'm preaching to the choir here, you're familiar with, but when I take this talk outside of this group, it's much less well understood. You know, from my perspective, I like to go beyond the crystal structure and look at the defects. A fundamental limitation of the crystallographic coding is it's only defined for crystals. And when I say crystals here, I actually say long-range periodically ordered structures. I know that the definition of a crystal includes non-periodic long-range ordered materials, but really, when we're talking about asymmetric units in unit cells, we're talking about the periodic case. So given that it's a crystallographic coding, it's kind of defensible that it's this way, but many modern materials don't fall into this category, and they're like the ugly children that are ignored by the community because they don't have this beautiful thing of being periodic. Something that I've noticed for a while, but Vitaly's really been leading publicizing this thing, is that it's not continuous. This is not a continuous coding, which means that you can make tiny changes in an atomic arrangement and it will result in a highly discontinuous change in the coding. So you move an atom off a special position and it moves from one space group to another. Even if that's a tiny change in the atomic arrangement, it produces this large change in the code. And in some sense, if you want to have a genomic code, things that are close structurally, you want them to be close in the code as well. This is actually the same point, but in a slightly different way. It's not a metric. So because of this discontinuity, structural similarity is not apparent in the code. Another huge one, again, that Vitaly's been pointing out, it doesn't respect isometry. but material properties do. So that's not 100% true. Like sometimes things will couple to a vector field or something, but on the whole, the properties of materials don't depend on kind of translational and rotational orientation of crystals, but they depend on the relative positions of the atoms in the crystal. And so if the structure and the properties respect isometry, we'd rather have a coding that also respects isometry. All right, so what are some alternative codings that we could explore? And again, you guys are familiar with this, but some stuff is actually, some thinking has gone into this outside the current group. So actually, as early as the 1980s, people wanted to do chemical informatics. They were thinking about doing genomic kind of things on structures. And what they needed is they needed material fingerprints. So they were already seeking these one-dimensional codes for structures back in the 1980s. And for molecular systems, a very important coding was called SMILES. These are SMILE strings. And you can imagine that you have here's morphine. Let's pick morphine, why not? And it's a molecule that looks like this. It has carbons and oxygens. They're all bonded together. And we want to come up with a one-dimensional fingerprint for this thing. This is a three-dimensional structure, but we want a one-dimensional fingerprint. So the SMILES formalism was this. You say, okay, I'm going to take this three-dimensional thing and turn it into a one-dimensional thing by like cutting bonds. So all of these bonds that went from continuous lines to dashed lines, these are ones that I've cut. I have my pair of scissors and I'm cutting them. And I cut it in a way that there's like a one-dimensional topology that I can follow all the way through this molecule here. Once I have that one-dimensional topology, what I can do is I can then write down a one-dimensional string. So I come up with a code which tells me basically kind of what the neighbors are. And I can't even remember what this coding is, but it goes O1, C2, C, paren O, C. So things that are connected without parentheses, you're just going along the chain. So O1 is this first oxygen is connected to C2, which is connected to this C. I don't know why that doesn't have a number on it. And then there's a paren because there's an oxygen hanging off the side and so on. And then this is a double bond. And so you can see this is actually encoding the structurally relevant aspects of this molecule in a one-dimensional string. And then I can store that in a database. And then I can search that database for, and then given this string, I can uniquely reconstruct this molecule. So it's not bijective though. So there's not a unique mapping from the molecule to this string. There's a unique mapping from the string to the molecule. So I could have initially chosen different molecules to snip, and then I would have ended up with a completely different string. But nonetheless, it's around, it's widely used. It's not going away. More recently, people have also wanted to start thinking about continuity, which is something that Vitaly is very interested in. So continuity means making, they call it materials cartography. This is also something that Vitaly's thought about. It's like making a kind of a two-dimensional map of a material space, where again, two atoms, two materials that are close to each other in this map are presumably somehow related. What these guys did back in 2015, and this one is not so heavily used. It's a kind of a paper that I just dug out of the literature. I'm not sure if it's really been adopted or widely used. But it's basically a way of getting this kind of cartography, this way of mapping materials. And the way that they did it is they said, what we're fundamentally interested in is actually the, not the structure per se, but the properties of the materials. So they did electronic structure calculations on all these materials, and they came up with descriptors of the properties. And I don't actually know what I mean here by simplices of structural components, so please don't ask me that question, but I presumably stole that out of the paper. I think I know what a simplex is, but anyway, so somehow or other they have this description and it allows them to make this map. So it's just to say that other people are trying to think in this direction, but it's still a minority of people. Okay, So these are existing fingerprints. The one that's my favorite is this atomic pair distribution function. And the reason is that I spent my entire career basically developing this as an experimental method. So very briefly, what is the PDF? So basically, it's the three-dimensional autocorrelation of the density distribution. So what I do is I define my material as a point cloud, where the atoms are the points and they exist in some Euclidean space. So I've got my 3D point cloud, and then I take the autocorrelation of that point cloud. So I convolve it onto itself, and that is the PDF, 3D PDF. So it's interesting for us because it's actually possible to measure it in principle. In practice, it's like slightly difficult to measure it, the 3D PDF, although we're actually getting better at it. But some things that we like about it is that actually this thing automatically respects isometry, because when you take the autocorrelation of the density, the absolute positions of the atoms no longer matter. It's only the relative positions of the atoms that matter. And so once you do this, you automatically have something that's isometry respecting. It's basically a map of the relative distances and vectors. Actually, in the 3D case, it's a map of the relative vectors between atoms. It's possible to compute in the forward direction. So given a three-dimensional point cloud, you can compute this object, and it's something that we can measure. So we like the PDF. These are the kind of experiments that we do to get it. So we have a nice, this $1 billion synchrotron, and we put our little bit of powder right here at the end of this tube, and this very intense X-ray comes down the tube, hits the sample, goes into the detector. This is a one-dimensional PDF. We get an isotropic signal coming out, and we integrate it, and we do corrections, and then we get our PDF. The one-dimensional PDF is the one that we deal with experimentally much more frequently, and this is where we have a sample which is made up of not a single crystal, but a lot of small crystals all randomly oriented. And so we do an orientational average of the 3D PDF. And so what you get is a one-dimensional function which has peaks at certain positions corresponding to interatomic distances. So if you sit on this atom and you go to this one, it's 1.4 angstroms away. On C60, it's a carbon-carbon bond, and here's a peak at 1.4. This is the second neighbor distance, and the second peak is at 2.5. This is the third neighbor distance across the hexagon, and it's at 2.8. And so these three peaks tell you that you've got hexagons in your material. Inside C60, you have also pentagons. In order to get this this sheet to roll up like this, you have to put fivefold rings in. That introduces curvature. And so then you have a mixture of 6 and 5-fold rings. And the 5-fold rings has the nearest neighbor distance and the second neighbor distance, but it doesn't have the third neighbor distance. So I can just do statistics on the relative intensity of the third peak to the second peak, and I can extract from that the relative number of 5-fold rings to 6-fold rings in my structure, for example. But I can also do the forward problem. I can do a double sum over every atom in this C60 molecule, and I can find where all of the interatomic distances are, and I can make a histogram, and this is a histogram. And the longest distance in C60 is across the diameter from this guy to this guy, for example. And it's a 7 angstrom ball. And so the last sharp peak is at 7 here. That's a 6 and that's an 8. So this is 7. And remarkably, this PDF here is a PDF of C60, but it came from this measured data set. So we took actually a powder of C60, we put it into an X-ray beam, we measured the intensity, we Fourier transformed it, and we got this thing, which is exactly the PDF of this C60. So it's kind of a miracle. You can see there's a little bit of intensity out here, and that's actually coming from the molecular packing. So I can equally well sit on an atom on this ball and look at an atom on this neighboring ball and find the interatomic distance. And what I see is that there's a broad peak peaked at 10, and 10 angstroms is the center-to-center distance of these two balls in this packing. And this is an FCC packing, and so the second neighbor distance is root 2 times 10, which is 14, and the third neighbor distance is root 3 times 10, which is 17. And so these three broad bumps here are telling us about the crystallographic arrangement of the balls in space. So not atom-atom correlations, but these are ball-ball correlations. And so just by sticking the powder in the beam, measuring the signal, Fourier transforming it, we get a huge amount of really rich information. So like, I would say what's not to love about the PDF? We should all love the PDF. So let's take a slightly more mathematical view of it. So this is my C60 ball, which we just know that we love. And I can say, okay, I'm going to represent it not as a continuous histogram now, but as discrete distances. So I'm going to assume that this is a point cloud, and these are located perfectly in space. There's no atomic motion that broadens them out into Gaussians. And so then I have a distance list. And so this distance list is the thing that I'm proposing for the genome, because it's a discrete list. It's something that's not a continuous function. It's a discrete set of distances. There are some distances that have higher multiplicities because of the symmetry of the thing. In principle, I could make a random point cloud, which this thing is actually a three-dimensional thing. It looks like a graph, but it's really three-dimensional point cloud. And then I extract the distance list from this, and here that they all have multiplicity 1, and they're all a different disks. So this is actually a generic graph, but in principle, any point cloud I could actually represent this way. And so this is an approach that you can start taking the distance geometry problem, which is taking something which has geometry, point cloud, decomposing it into distances, putting those onto a graph where the edges contain the interatomic or the interpoint distance, and the nodes of the graph contain attributes of the points or the atoms, let's say. So I'm not going to talk about distance geometry problem, but in principle, there's a kind of mathematical formalism for it. So here's my big claim then. The PDF is a 1D discrete list of quantities that codes for the 3D structure. So let's make the PDF to be the materials genome. This is the distance geometry problem, which some work was done by Phil Duxbury, who was a colleague of mine at Michigan State University. We did it together, and then Carly Lavore and Leo Liberty, and then Vitaly has really also picked this up or kind of done it independently, essentially, where he's actually computing these isometric invariants of the DGP. Vitaly's going to tell you about this if he hasn't already, but I like very much this slide because it actually, he has it laid out exactly what the problem definition. Like when I talk to my students, I say finding solutions is easy, defining the problem is what's hard. And this is actually, for me, a very beautiful and elegant definition of the problem. So we want invariant, so we want no false negatives. So that's what the definition of the invariant is. We want it to be complete, so there's no false positive. So this would be, in some sense, a generic definition of what we want for our code. We want it to be a metric. I already discussed this. So you want the code, things that are close in structurally or in terms of properties, they're close in the code. And you want it to be continuous. And the other nice little one at the end is we haven't really done much if we come up with a coding that is so difficult to compute that it's useless. So so Vitaly, thanks for this. I like this very much. So he's been pushing this continuous crystallography. I'll let him talk about it. But this is a restatement of what I just said. And then, so I thought I had another slide here, which is some of his invariants that he'd come up with. So things like, is it TPP and ADP? There's a nice list of them.
Do you mean PDD, point wise?
PDD, yeah. So they're not. So Vitaly's a mathematician. He came up with them on his own, or his group did. But they're somehow closely related to this PDF and different ways of defining the PDF. So I like to actually also think about this mapping and to include Vitaly in the PDF family. Okay. So this is the part where the talk is going to diverge a little bit from last year, which is, okay, I started thinking then to begin with, it was just a fun thing. I wanted to say, look, materials don't have a genome, but then it's like, well, what if they did have a genome? What would that genome be? And then, oh, it could be the PDF, why not? But you can then take this idea a little bit further and you can say, look, well, let's look at other things that a genome does. How does that map onto the materials case? So one thing that you need for a genome to be useful is you need a ribosome. Ribosome is some cellular machinery. It's the thing inside the cell that converts the code into the structure. And so it takes the ribosome takes the DNA and it reads it, and then it figures out which molecule to make, and then it makes the molecule. So that's how our bodies work, basically. These proteins are all enzymes, they do chemistry. When a cell needs some particular chemistry to be done, it needs to manufacture the protein that does that chemistry. The ribosome manufactures it. When that chemistry is done, it needs to break that down into its constituents again. So the ribosome is the machinery that builds the 3D structure from the 1D code. So if we're going to use the PDF as our materials genome, we need a materials ribosome. So we need machinery that goes from the code to the 3D structure. And so that's a structure solution from PDF. So we actually did some work on this because we measure PDFs, and we always wanted to have a machine where you give it as input a PDF, a one-dimensional pair distribution function that's measured, and it gives you as output the 3D structure. And we basically solved the problem. We actually had a paper in 2006 in Nature. So somebody thought it was worthwhile. And this was actually a cluster build-up algorithm that was based on European soccer leagues. So what we did is we had a bunch of clusters of different sizes, and we competed them against each other with a scoring function. The scoring function was how close they were to the true structure. And then clusters that competed well, we promoted them, which meant that they could buy new players, so they grew. And then clusters that performed poorly, we relegated them. And so they were playing in a lower league, and so they had less money, so they had to sell players. So we had a mechanism for growing clusters that were performing well and shrinking clusters that were performing poorly. And it's actually a really efficient algorithm. We could actually solve structures from PDFs. Now, if that had been the whole story, then I wouldn't be sitting here now. I'd be extremely rich and famous, and not talking to a bunch of mathematicians on Zoom. But unfortunately, it actually didn't solve the problem as we had hoped it would. And it was largely speaking, it did actually work. We did solve a bunch of structures. Here's a list of a bunch of structures that we solved really from experimental data. But if you look at them, they're all small structures that are kind of trivial to solve by many other methods. And when we got to more complicated structures, we ran into a problem. So this was a little bit of analysis that Pavel Juhas, my postdoc at the time, did on this. So what we did is we went to a structural database and we pulled out a bunch of structures. And I won't go completely into the details, but roughly speaking, they were molecules, and they had size from 0 atoms to 160 atoms. So they went from small to big. And what we found was that sometimes LIGA succeeded and sometimes it failed. So these stars were the ones it succeeded on, and the crosses are the ones it failed on. And you might think that, oh, it's going to fail on the big ones and it's going to succeed on the small ones. But what we found was that actually it could succeed on really big ones. And sometimes it would fail on very small ones. So the size of the cluster was not the issue. But what Pavel did is he put it onto this plot, which separated it into 2 axes. And the second axis is the number of unique distances on the cluster. So you can imagine that if you have some number of atoms like 40, if it's a very high symmetry, if it's forming a very high symmetry cluster, it's going to do that with a small number of unique distances. But if it's forming a very low symmetry cluster, it's going to take a large number of unique distances to specify the cluster. And so basically, when you go up on this direction, you're going down in symmetry. And if you have no symmetry, you have a completely random cluster, you're going to follow this curve here, which is n into n minus 1 over 2, which is the number of degrees of freedom in that problem, that pairwise problem. So everything has to be below this blue curve. And what you can see now is actually, it doesn't divide by big and small, it divides by, basically there's a line of death here. If you're below that line, we can solve you. And if you're above that line, we can't solve you, regardless of size. And so it's something about structural complexity. And in the end, we felt that it was something to do with an information theoretic problem, that when you have a large number of unique distances, if you could measure that with perfect resolution, you would have more information in your input data than if you had a small number of unique distances. But when you measure it with finite resolution, these distances start to overlap. So I can quickly go back to this generic graph. This graph, this histogram down here has many more distances than that one. So this one down here has more information. But if I do a measurement of this with a broad resolution function, then I'll end up with just a horizontal line here, largely speaking, a horizontal line. Whereas here, I'll have one which has a lot of ups and downs in it. That's exactly what the data look like. So if I go back to this, actually, I have these inverted, sorry about this, but the ones that have low symmetry, their measured PDF looks like this. And the ones that have high symmetry, their measured PDF looks like this. And so it turns out that actually it's not a problem of the algorithm, that the algorithm can't solve those. It's a problem, the information content in the data is insufficient to constrain a correct solution. Okay, I thought, okay, so I was gonna, well, something that we've been doing now is actually trying to apply machine learning to this. And what machine learning does is it actually gives prior knowledge to the machine learning model about possible structures, and it's actually starting to work, but it looks like I took those slides out. All right, so it's appropriate though that I switch, because we're running to the end of the talk, and talk about the two kind of new concepts. So the one I want to switch to now is structure definition problem. So something that comes out of this structure solution issue is the following. What we want to do is we want to have our box. We give the box as an input of 1D PDF. It gives us an output as a structure. And we want to ask the following question. Did it solve the structure correctly or incorrectly? So we want to take, let's say we have a ground truth. We knew what the structure was that gave that PDF. We give that PDF to our black box. The black box tells us what the structure is it thinks it is. And we want to answer the question, is it the right structure? In order to do that, we have to have a way of mapping the given structure on top of the ground truth structure, right? So how do we do that? And so, of course, Vitaly says, well, you use DPP, you use my continuous crystallography metrics, and so on and so forth, right? But what I realize is that actually this is a problem which is not a mathematical problem. It's a kind of a sociological problem in the business. So the question is, what is a structure? And it kind of depends who you ask. If you ask a chemist, they'll tell you that they'll talk about an FCC structure, and they'll say that's a structure. But you can have iron as a FCC iron, and you can have FCC nickel. But according to Vitaly, FCC nickel and FCC iron are different structures because they have different interatomic distances. But according to the chemists, those two things have the same structure. So there's an ambiguity in what we mean by structure. And so I just want to, well, luckily we have International Union of Crystallography, and we're in the business of defining things. So how does IUCR define crystal structure? So only a room full of mathematicians could love this. this is the official august IUCR definition of crystal structure. It's really concise. A crystal structure is a crystal pattern consisting of atoms. So there you go. Show that to a chemist and they're not going to know what the hell you're on about. So, well, okay, we have to click on crystal pattern because clearly this is a nested definition. So this is crystal pattern. So the definition of crystal pattern is an object in the n-dimensional point space En is called an n-dimensional crystallographic pattern, or for short, crystal pattern, if among its symmetry operations there are n translations, the translation vectors t sub 1 up to t sub n, of which are linearly independent. And all translation vectors, except the 0 vector, have a length of at least d greater than 0. So functionally, this is a complete, so this is rigorous, it's mathematical, it's well-defined, but functionally it's completely useless, right? Because it doesn't help me answer the question of like whether FCC iron and FCC nickel are the same structure or not, right? Well, actually what this tells you is that they're not, because they're going to have different sets of translation vectors. But yeah, it's functionally not a very helpful definition of structure, I would say. So there's also the kind of the sociological definitions, which is something to do with what chemists think. So what's a chemist definition? I actually tried to use Wikipedia to find their definition. And when you go to crystal structure on Wikipedia, it just starts telling you crystallography. It doesn't say the definition of a structure is. So maybe in some chemistry textbooks it's there. But basically, the chemists actually, they take this crystallographic view. They define a structure based on the space group classifications. So they say structures that change without breaking symmetry are the same structure. But as soon as you break symmetry, you have a new structure. So if the chemist's definition is the correct one, or a functionally useful one, then our continuous crystallography is great, because we can say when two structures are the same, because their invariant computes to the same number. But if the invariant computes to a different number, we can't say whether those are the same structure or not from the chemist's point of view. Okay, let's do a little thought experiment here. Think about thermal expansion. So we'll take a material, could be nickel or chromium or something, and we warm it up. When we do that, because of thermal expansion, the unit cell expands, but it does it typically without breaking symmetry. So if it's a cubic unit cell, all 4 axes expand at the same rate, it's still a cubic cell, right? So from the chemist's point of view, the structure hasn't changed. In fact, the chemist would think you're a complete lunatic if you say that I take nickel, I warm it up 10 degrees, and now it's a different structure. They would laugh you out of the room, right? So from their point of view, the structure hasn't changed. From the continuous crystallography point of view, the structure has changed, because its set of interatomic distances are all different, right? And then what about from a real kind of materials point of view? Well, actually, the structure has changed from a materials point of view, because all of the bonds have changed their length, and then the quantum mechanical wave function overlaps have changed, which means the quantum mechanical spectrums change, which means that the properties have changed. It's metallic, its resistivity has gone up, and so on, right? So it's just this thing that actually, yeah, it doesn't make sense to say that you warm up a piece of nickel 10 degrees and it's got a new structure. That's, from a chemist's point of view, doesn't make any sense. But the chemists have also a lot to answer for too, because here's something that can happen as well, which I think is also kind of slightly ridiculous. I can make a minuscule phase transition. So I can take a material, the same material, nickel, let's say, and I can pick one atom in the unit cell that sits on a special position, I can move it by a tiny amount so that it moves off the special position, right? Then I've actually changed the space group. And so from the point of view of the chemist, the structure has changed. The chemist would say that's undergone a structural phase transition. It's a new structure. But if I computed the continuous crystallography measure, should that be DPP? I could tell you.
Simon, so it's very similar to PDF.
Yeah.
PDD, pointless distance
PDD.
distribution.
That's how I'll remember it. It's very similar to PDF. Yeah,
Yeah, very
I don't know
similar.
why I have such a hard time with this. I'm so sorry. But whatever your continuous crystallography measure is that you use, because you just took one atom and you moved it a tiny amount, the crystallography measure changes by a tiny amount, and that's appropriate because the properties are going to change also by a tiny amount. So this is good. But the chemists say, no, that's things changed its structure. So the chemists also need to think, we need to think carefully about if we have a good definition of structure. So I don't have an answer to this problem, but I'm just raising it. I'm just raising it as something that we need to discuss. And Greg is on this call, and he's deeply involved in IUCR now. Greg, I'd love to get your views on this and think about how to take it forward. But as a community, we need a more practical definition of what it means for two structures to be similar. I mean, I think it's straightforward to define two structures as being the same. But what does it mean when they're not quite the same? So I just want to leave you with that. Oh, these are the slides that I had I'm not going to go through them now. Everything got out of order, sorry about that. Oh, okay, here you go. Okay, so in the last few minutes then, this was the structure definition problem. The last thing I want to think about too is, again, we were playing this game of, oh, materials don't have a genome, but what if they did have a genome? What are some other aspects of genomics? And a really important one is obviously heredity, right? So we're human beings, and presumably we've evolved to this point through natural selection. And the way that natural selection works is that we continually get better over time. And the way that we continually get better over time, or more fit to our environment, let's say, is that small, tiny stochastic mutations occur to the gene code, and that these small local mutations result in small changes in the resulting structure. And sometimes that small change in the resulting structure makes that organism more fit to their environment. And then over time, that mutation will be absorbed into the genome of the larger population, and so on. So having the ability to make small local stochastic mutations to your genome allows this continuous improvement through natural selection. So in principle, we could make better materials. We could realize the vision of Materials Genome Initiative through this kind of way of locally mutating our materials genome. Okay, so I started out by saying the PDF is a materials genome, and what I've realized is the PDF is a terrible genome for materials. And the reason is because you can't make small local mutations to it. Basically, if you take one of the distances in your distance list and you mutate it, you change its value a little bit, it results in a graph which won't embed in E3. In fact, it results in an invalid PDF. So if you take one atom and you move it, you update many, many entries in your codon, and they're not even localized within the codon. So you can't make a small stochastic modification to your codon, and it will result in a valid structure. So I guess the final bit of my talk is, sadly, because I love the PDF so much, it's actually completely useless as a materials genome. So let me just, I think I've used up my time. So the summary is, we should use the PDF as a materials genome, but actually we shouldn't. So let me finish there. And this is a Zoom style picture of my group, and I'd like to thank all of them, and I'd like to thank also my collaborators and the funding and so on. And with that, yeah, I'm happy to take some questions.
Thank you very much, Simon, for your great overview. Please thank Simon. Thank you very much. Oh, please keep the slides.
Oh.